Hello everyone, today I will share our recently published study in iMeta, entitled Patient-derived organoid-immune co-culture integrated with multi-omics analysis reveal mechanisms of immunotherapy resistance in urothelial carcinoma. FGFR3 is one of the most common oncogenic driver mutations in urothelial carcinoma, and its activating mutations have been closely associated with tumor immune suppression. However, most previous studies have relied on conventional cell line models. As a result, there remains a lack of patient-derived systems to fully investigate the underlying mechanisms. This study integrates patient-derived urothelial tumor organoids and immune co-culture systems with multi-omics analyses to investigate FGFR3 function. FGFR3 mutant tumors display a cold immune microenvironment associated with poor immunotherapy response and prognosis. Mechanistically, the FGFR3 inhibitor remodels this microenvironment via the FGFR3 STAT5 IRF2 axis, enhancing anti-tumor immunity. We first demonstrated in clinical samples that FGFR3 mutations are closely associated with an immune-depleted tumor microenvironment, characterized by reduced infiltration of immune cells, along with decreased T-cell cytotoxicity and increased exhaustion. To model tumor-immune interactions, we established patient-derived urothelial tumor organoids and immune co-culture systems, which preserve tumor features and enable antigen-specific T-cell activation. This provides a clinically relevant platform to study tumor microenvironment. Targeting FGFR3 reduces the proportion of exhausted T-cell subsets, while increasing GNLY-positive cytotoxic T lymphocytes and NK cells. This shift is accompanied by upregulation of key effector molecules, such as GNLY, and enhanced NK cell-mediated cytotoxicity. We further explored how activating FGFR3 mutations drive immunosuppression. IRF2 was identified as a key upregulated transcription factor, binding interferon-related gene promoters and acting downstream of FGFR3 signaling. Functionally, STAT5 inhibition suppressed interferon signaling, as erdafitinib restored GZMB secretion. Overall, erdafitinib activates the STAT5 IRF2 axis to reverse immunosuppression and promote anti-tumor immunity. Chemokines regulate tumor immunity by mediating immune cell recruitment. We therefore assessed chemokine expression in the co-culture system and integrated these data with IRF2 cut and tag results. Erdafitinib treatment significantly upregulated chemokines such as CCL4, XCL1, and XCL2. Notably, CCL4 is a well-established chemokine that efficiently recruits NK cells and cytotoxic T cells, playing a central role in promoting anti-tumor immune infiltration. Cut and tag analysis further showed that IRF2 directly binds to the promoter regions of these chemokines, confirming its role as a key transregulator. Together, these results support a STAT5 IRF2 CCL4 axis that drives chemokine expression and promotes recruitment of anti-tumor effector T cells. To validate the IRF2 CCL4 axis in vivo, we generated MB49 bladder cancer cells expressing the FGFR3 mutant and established a syngeneic model in mice. Erdafitinib treatment increased IRF2 and CCL4 expression and enhanced infiltration of GZMB-positive cells and NK cells, consistent with in vitro and patient data. These findings indicate that FGFR3 inhibition drives the recruitment and activation of CTLs and NK cells both in vitro and in vivo via the STAT5 IRF2 CCL4 axis. Finally, we evaluated the potential of combining FGFR3 targeted therapy with immunotherapy. In co-culture system and mice model, both monotherapies inhibited tumor growth, while the combination produced significantly stronger anti-tumor effects, with better control of tumor progression and weight loss. Thus, FGFR3 targeting preconditions the tumor microenvironment, enhancing responsiveness to immunotherapy and yielding synergistic anti-tumor effects both in vitro and in vivo. Overall, our findings demonstrate that activating FGFR3 mutations drive an immunosuppressive microenvironment in urothelial carcinoma, limiting immune cell infiltration. Importantly, combining FGFR3 inhibitors with anti-PD-1 therapy produces synergistic anti-tumor effects, highlighting its translational potential for improving immunotherapy outcomes in FGFR3 mutant tumor.